Yes, sir.
ಓಕೆ ರಾಘವೇಂದ್ರ ಸರ್, ಶುರು ಮಾಡೋಣ.
Yeah, definitely.
ಓಕೆ ಓಕೆ. ಎಲ್ರಿಗೂ ನಮಸ್ಕಾರ. ಗ್ರೆಗರ್ ಮೆಂಡೆಲ್ ಅವರ 200ನೇ ಹುಟ್ಟುಹಬ್ಬದ ಆಚರಣೆಯನ್ನ ನಾವು ಇವತ್ತು ಒಂದು ವಿಶೇಷ ಉಪನ್ಯಾಸದ ಮೂಲಕ ಮೈಸೂರು ಸೈನ್ಸ್ ಫೌಂಡೇಶನ್ ಮತ್ತೆ NCTS ಎರಡು ಜೊತೆಯಾಗಿ ಆಯೋಜನೆ ಮಾಡ್ತಾ ಇದೆ. ಇದರ ಒಂದು ಉಪನ್ಯಾಸವನ್ನ ಆಯೋಜನೆ ಮಾಡಬೇಕು ಅನ್ಕೊಂಡಾಗ ನಮ್ಗೆ ಮೊದಲು ನೆನಪಾಗಿದ್ದು ಡಾಕ್ಟರ್ ರಾಘವೇಂದ್ರ ಅವರು. ಡಾಕ್ಟರ್ ರಾಘವೇಂದ್ರ ಅವ್ರನ್ನ ಕೇಳಿದಾಗ ನಾನು ಅತ್ಯಂತ ಸೂಕ್ತವಾದಂತಹ ಒಬ್ರು ಸಂಪನ್ಮೂಲ ವ್ಯಕ್ತಿಯನ್ನ ನಿಮ್ಗೆ ಆಯ್ಕೆ ಮಾಡ್ಕೊಡ್ತೀನಿ ಅಂತ ಹೇಳಿ ಡಾಕ್ಟರ್ ನಲ್ಲೂರ್ ಬಿ ರಾಮಚಂದ್ರ ಸರ್ ಅವ್ರನ್ನ ಪರಿಚಯ ಮಾಡಿಕೊಟ್ರು. ನಿಜವಾಗ್ಲೂ ಬಹಳ ಖುಷಿ ಆಯ್ತು ಅವ್ರ ಜೊತೆ ಮಾತಾಡಿ. ಮೈಸೂರು ಸೈನ್ಸ್ ಫೌಂಡೇಶನ್ ಇಷ್ಟು ವರ್ಷಗಳು 10 ವರ್ಷಗಳು ಕೆಲಸ ಮಾಡಿದೆ, ಸರ್ ಅವರ ಒಂದು ಉಪನ್ಯಾಸವನ್ನ ನಾವು ಆಯೋಜನೆ ಮಾಡಿಲ್ವಲ್ಲ ಅಂತ ನಿಜವಾಗ್ಲೂ ನನ್ಗೆ ಬೇಜಾರಾಯ್ತು. ಅದೇನೋ ಅದೃಷ್ಟನೇ ಸರಿ, ಡಾಕ್ಟರ್ ರಾಘವೇಂದ್ರ ಅವರ ಮುಖಾಂತರ ಸರ್ ಅವರ ಪರಿಚಯ ಆಯ್ತು. ಅವರು ಇವತ್ತು ನಮ್ಮ ಈ ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ಉಪನ್ಯಾಸವನ್ನ ನೀಡಲಿಕ್ಕೆ ನಮ್ಮ ಜೊತೆಗಿದ್ದಾರೆ. ಅವರ ಒಂದು ಪರಿಚಯವನ್ನ ಡಾಕ್ಟರ್ ರಾಘವೇಂದ್ರ ಅವರೇ ಮಾಡ್ಕೊಟ್ರೆ ಅತ್ಯಂತ ಸೂಕ್ತ ಅಂತ ನಾನು ಅವರನ್ನೇ ರಿಕ್ವೆಸ್ಟ್ ಮಾಡ್ಕೊಂಡೆ. ಹಾಗಾಗಿ ಡಾಕ್ಟರ್ ರಾಘವೇಂದ್ರ ಅವರು ಕೂಡ ನಮ್ಮ ಜೊತೆಗಿದ್ದಾರೆ. ಡಾಕ್ಟರ್ ರಾಘವೇಂದ್ರ ಸರ್, ಪ್ಲೀಸ್ ಇಂಟ್ರೊಡ್ಯೂಸ್ ಅವರ್ ರಿಸೋರ್ಸ್ ಪರ್ಸನ್.
ಎಲ್ಲರಿಗೂ ನಮಸ್ತೆ. ಇವತ್ತು ನಿಜವಾಗ್ಲೂ ಬಹಳ ಖುಷಿ ಆಗುತ್ತೆ. ನನ್ನ ಸುಕೃತ NBR ಸರ್ ನಮಗೆ ರೋಲ್ ಮಾಡಲ್. ಆ್ಯಕ್ಚುಲಿ ವಿಜ್ಞಾನವನ್ನು ಎಷ್ಟು ಸೀರಿಯಸ್ ಆಗಿ ತಗೋಬೇಕು, ಅದಕ್ಕೆ ಎಷ್ಟು ನಾವು ಆ್ಯಕ್ಚುಲಿ ಕಾನ್ಸಂಟ್ರೇಟ್ ಮಾಡಬೇಕು, ಡೆಡಿಕೇಟ್ ಮಾಡಬೇಕು ಅನ್ನೋದನ್ನು ಅವರು ತುಂಬ ಚೆನ್ನಾಗಿ ಅವರ ಎಲ್ಲ ಸಂಶೋಧನೆಗಳ ಮೂಲಕ, ಅವರ ಲೇಖನಗಳ ಮೂಲಕ ಸಾಕಷ್ಟು ನಿರೂಪಿಸಿದ್ದಾರೆ. ಅವರು ಸಾಕಷ್ಟು ಒಬ್ಬ ಉತ್ತಮ ಟೀಚರ್ ಆ್ಯಂಡ್ ಈಸ್ ಆಲ್ಸೋ ಎ ವೆರಿ ಗುಡ್ ರಿಸರ್ಚರ್. ಸೊ ಐ ಆಮ್ ಹ್ಯಾಪಿ ಟು ಇಂಟ್ರೊಡ್ಯೂಸ್ Dr. NB Ramachandra sir, who served as, for long years, he served as professor in Department of Zoology, and presently is working as UGC BSR faculty fellow, Department of Studies in Genetics and Genomics. And I think Department of Studies in Genetics and Genomics is the brainchild of Professor NB Ramachandra sir. It's doing wonderful work in collaboration with so many collaborations are there with abroad. And happy to mention that the Department of Studies in Genetics and Genomics is located in Manasagangutri, University of Mysore. And he is also serving as professor, honorary professor in Sri Satya Sai University of Human Excellence, Kalaburgi. And he is also serving as consultant in Human Genetics Unit, All India Institute of Speech and Hearing, located in Mysore. And is a chairman of Institutional Ethics Committee, Mysuru Medical College and Research Institutes in Mysuru. And very interestingly, he is updating his, this is what actually appreciating regarding, so he's getting updated every things. And he has contributed two courses, if I believe so. And one important course is Human Genetics, for the massive open online courses which is still running under the leadership of NB Ramachandra sir. And it is funded by MHRD, Ministry of Human Resource Development, New Delhi. And if we recall his service, he served as a professor and founder chairman of the Department of Studies in Genetics and Genomics, University of Mysore during 2015 and 2020. During 2007 and 2015, he was the professor and coordinator of MSc Genetics, again, Department of Studies in Zoology, University of Mysore. In 2001, he worked as visiting professor for University of Innsbruck, Austria. In 1999-2000, he was worked as senior research associate 2 at University of California, Los Angeles, USA. In 1999 and 2009, during this period, he was the reader, associate professor actually in the present designation, a reader in zoology, Department of Studies in Zoology, again University of Mysore. And he was a lecturer in the same department during 1992 and 99. And he was a postdoctoral scientist at McMaster University, Canada during 1992. And during 89 and 90, he was the CSI research associate at the Department of Studies in Zoology, University of Mysore. During 88 and 89, he was the ICMR Research Associate at NIMANS, Bangalore. During 82 to 88, he was I think he obtained a PhD in zoology in genetics from the same University of Department of Studies in Zoology, and he obtained his master's degree in zoology from the same Department of Studies in Zoology, during 1980 and 82. So this speaks of his continuous thrust for the knowledge update, and he is into teaching and he is into research, and all these years, this progress shows is very active in research and academics. And regarding his research contribution, he has guided 29 students for PhD. That's a great work, sir. And three are presently working in the field of Drosophila genetics and evolution, and human genetic diseases and genomics. And he has contributed more than 300 research articles in the national and international conferences he has attended, and he has also contributed research papers. And his publications goes to the maximum 279. He has contributed in terms of research articles, gene sequences he has deposited, research notes and chapters in books in national and international journals and publications. And he has academic, administrative, and professional responsibilities to the tune of 110. And a very important thing is entire his career, he has completed several projects, and if it goes to 17, and it was funded by Department of Biotechnology, Department of Science and Technology, Council for Scientific and Industrial Research, Indian Council of Medical Research, University Grants Commission, and he has also a collaboration research work at international level in collaboration with Canada. And he was the life member, I think he's serving as a life member of many national and international professional societies. Very interestingly, he has a H index that shows how much of his research contribution has been taken by other scientists all over the world. It goes up to 25. And he has I-10 index of 64, and total citation goes up to 2,100. That's a great contribution for the young researchers and those who are working in the different fields of genetics. And of course, having put in a lot of work on research and teaching, he has been recognized with so many awards. In 2021-24, editorial board of Annals of Genetics, international open access peer reviewed journal published by Mediterranean journals. It's a great recognition. And in 2020-21, and he was the honorary fellow of Karnataka Science and Technology Academy. In 2021, Shisharu Mechida Guru Award was given to him by Negala Yogi Samaja Seva Trust of Mysore. In 2019, he was the NAC Review Expert Member. 2017, FASC elected Fellow of Indian Academy of Sciences, Bangalore. Kindly note, this is only given to few people in India, and one such is with us, N.B. Ramachandra sir. In 2016, University of Mysore Golden Jubilee Foundation Award he has obtained. In 2015 and 16, he served as local secretary for one or third Indian Science Congress, which is held in 3rd January 2016. And very interestingly, the Karnataka state highest recognition, Dr. Raja Ramanna Scientific State Award, was given to him in 2014. And 2013 Engineers State Award was also endowed on him. So in 2015, Professor G.K. Manna Memorial Award was obtained in 2015. In 14-15, he was also the Indian 102nd Indian Science Congress held at University of Mumbai, ISKA, Kolkata. So he has also participated in that, and he has also obtained award from the Vision Group on Science and Technology for his research publication. In 2018, he was the editorial member of the journal Zoological Research, Chinese Academy of Sciences, China. In 2007, he was endowed with, very interestingly, because early, I think, in his research career, Sir C.V. Raman Young Scientist Award for Life Sciences. That's again from the Karnataka state government. And his list of achievements is going on the several pages. It runs to several pages, and it is been actually reduced for the time sake. And it is honored to introduce you, sir, for Mysore Science Foundation. I'm really happy that this session will give more insights into what is genetics actually Mendel has given and what genetics is happening in the present scenario. Welcome you, sir, once again, N.B. Ramachandra, sir.
Thank you, Dr. Raghavendra and Santosh, for inviting me and giving this opportunity to share and to exchange my view regarding the genetics, what I learned and what I've done, some aspects what I am going to cover here. And it's a great honor our own local people are giving this opportunity to share my thoughts. ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. I will going to cover mainly Mendel's and his contribution towards the society, how these Mendelian principles are helping with the helping and with the society for the human better health care. So I hope my slides are moving, right, Raghavendra?
Yes, sir. Yes, sir. Yes, sir.
So this is the outline of my talk. I hope I will cover this in 50 to 60 minutes of this. So I'll just briefly cover about Mendel's work and extension of Mendelism, and particularly the human genetics and diseases, and few aspects of the genomics, and I try to conclude with the thumbing information that how important the genetics is today world. Gregor Mendel is the father of modern genetics, was born on 200 years ago on 20th July 1822. All over the world, the scientists, contemporary scientists are celebrating the Mendel's contributions and remembering the Mendel. I think this is one of the unique contribution, very humbly, honestly done by the Mendel. Mendel started the experiments with a pea as an Austrian monk, and his meticulous, systematic experiments made him to think that some factors, determinator, are passing from generation to generation, from parents to offspring. That was the idea he put forth. He was the first person to lay the mathematical foundation of the science of genetics. Now it's called Mendelism. Mendel, to do experiments on pea plants, he got motivation from his, there are many motivations. One is he was studied the biology. Along with the biology, mathematics also he has studied, and the physics at the University of Vienna. That has greatly helped later for his interpretations. Along with that, many times our environment is also very important. He had a very good friends, and 50s and 60s. The NAB, he was a sheep breeder. He was discussing with the Mendel how the new breeds or domesticated breeds can be produced, and all the details they were discussing. He had a good idea about the breeding and the hybridizations. Another friend now is a plant reproductive biologist. He was explaining how the reproductive systems of plants, pollen grains, and how the whole the inflorescence, how the self-fertilization takes place, or for cross-fertilization, how one has to do that. He is to discuss that. This is another contribution for his motivation. And he was teaching there mathematics to the school children, and all this has helped him to establish a consistent, rigorous methodology in breeding experiments. So many scientists this month in different articles have quoted that he is a very humble and a very curious person, and did his experiment with a curiosity and a very systematic manner. Mendel did the cell pollination of the plants. He studied all over for almost like 8 years, 29,000 pea plants. He established 34 kinds of the varieties of the Pisum setabum. And for 2 years of his breeding experiments, he could able to establish 22 purebred lines. Out of that, the interesting what we selected where he could able to reproduce his results are the 7 lines of the pea, which are different from each other for 7 distinct traits. This is the foundation for all the work, whatever he did and whatever he repeated and finally concluded as a law of the principles of inheritance. So contemporary understanding for the Mendel of that how the factors, how the characters are inheriting generation after generation. One of the principles were known at that time from the Aristotle believed that is the blending hypothesis, wherein the genetic material contributed by 2 parents mixes in a manner analogous to a way blue and yellow paints blend to make a green. So that means and the next progeny will blend and will have a character of both the parents. And another his observation made that he was not getting the blended the hypothesis support for the blended nature, but his experimental data could able to make something like a determiner or a factor is responsible and passing from parents to the offspring as a discrete heritable unit. That is the one making all this character. So he prepared that, he thought in that direction and tried to interpret all his data under this hypothesis, that is discrete units or particulate hypothesis. So he simply crossed the contrasting homologous individuals. Only one form of the trait will appear in the F1 generation, and that trait he called it as a dominant. The one which has masked is called as a recessive. And always whenever the seven lines whatever taken, he tested all of them out. All of them agree with this. Then he said that the character, the trait which is seen in the F1 generation, they are called dominant. The second hypothesis that the law we put forth was that when any individual produces gametes, because all this the knowledge he has from his friends that the plant reproduction, the copies of the gene or the factor separate so that each gamete receives only one copy, and a gamete will receive the one allele or the other. So every gamete will get a one copy, and that means a genetic determinant or the factor of a specific trait is passed on from one generation to next generation as a unit, as a discrete unit, without any blending of the unit. This he has repeated several times to make this kind of the statement and finally hypothesis, and they become the law. Even today this works, and his experiments he did in all the 7 lines of the contrasting characters where he established as a dominant as a recessive characteristic features, and also he counted the F2 progeny very meticulously, recorded them, and all this has been published in his article. All of them are showing nearer to 3:1 ratio. So his mathematical knowledge, his meticulous experiments make that something taking that the characters are not blended in F1 generation, they segregate to a gametes and produce the independent one, the progeny there. So he also these 7 the characteristic feature he could able to take 2 contrasting characters together and in the plants and cross them also, try to look for that if these contrasting characters are going together or separate or they are independent. So he did the experiments. He found that parental characteristics can be split and non-parental combination can be obtained. That means the parental characteristic, whatever is there in the plant, they will not go together. They can also split. He could able to then see them in 9:3:3:1 ratio. And the hypothesis what he put for that in a dihybrid or two characteristic cross, that is, if they are dependent, then they go together. They have to show 1:3 ratio. If they are these characters, two character contrasting characters are independent, they can produce the 9:3:3:1 ratio. And this is he repeated this in all the 7 lines, and that has made him very strong. All the monohybrid cross, a dihybrid cross, a law of segregation, law of dominant, law of independent assortment. He repeatedly getting the same kind of nearer to the same kind of ratio. That was, he was strong enough. This, I'm hitting the right kind of the hypothesis, right kind of principles, and then that made him to compile all the work and present his work, experiments in plant hybridization in 1865, February and March, at the Brune Natural History Society. He was excited to share his the experimental data that he presented, but not much encouragement, not much discussion, because he's bringing biology with the mathematics together. Many people are not that happy, not able to understand how to integrate that also. So to summarize what Mendel did, the data is that traits are distinguishing characteristics that are inherited. Traits are inherited as a discrete unit. The traits can be validated by backcross and test crosses, which give the expected ratios. And Mendelian inherit reflects the rules of probability. He calculated the probability, and the same manner the results are going together in that. And the approach what he made, the approach was experimental and also quantitative. He collected enormous the progeny of the F2 generation and the F1 generation that has helped him to make this. Therefore, genetics is the study of biological inheritance and patterns and variations. So Mendel laid the groundwork for the genetics with his 8 years of work with repeatable experimental data. So in the beginning, in the Nature editorial in the July 21st, they write about the celebration of Mendel's 200 years birthday, true legacy of Greg Mendel, careful, rigorous, and humble science. And their quote, the Mendel's paper's last paragraph, that I quote that, the validity of the set of laws suggested by Paisum requires additional confirmation. Thus, a repetition of at least the more important experiments would be desirable. This was the statement made by the Mendel end experiment. That means he was very careful and meticulous and humble, that whatever the data is collected, he made the other contemporary scientists to repeat that in other model organisms. So that's why they conclude this editorial that Mendel, as a careful scientist, cautious, patient, and committed to data, allowed him to make discoveries that have stood the time of test of time until even today. So this has generated the birth of the genetics in 1865. However, the contemporary scientists from this 35 years up to 1900, they were discovered the other contemporary like chromosomes and the chromosome movements in the cell divisions, and the others are handling better the mathematics, and also accepted the wisdom. That time accepted wisdom was that inheritance could not be studied in a scientific or statistical manner. They no one has appreciated what Mendel said in the ratio, and that was the wisdom that time had. However, Mendel's work was rediscovered in 1900 after a period of 35 years of gestation period by three independent plant scientists called Currens, Hugo de Vries, Tushmar, and all are the European scientists. They contributed independently. They read the Mendel paper, and their data also contributed what the Mendel said, and that's they are given the rediscovered, the title rediscovered Mendelism, and they appreciated the Mendel already 35 years ago. He made this hypothesis, that the theory. That was when they published this article, and Bateson was influenced very well in England and USA. He promoted the Mendel's work, and Mendel's idea provided what appeared to be the first comprehensive concept of heredity. He was convinced the contemporary scientist, and William Bateson coined the term genetics in 1905, and 1909 he was so enthusiastic to make the contemporary scientists to understand what Mendel said in his article. He translated the German text into an English text like Mendel's Principles of Heredity, and that is that has established genetics is a discipline of biology, is a science of genes, heredity, and variation in living organism. Am I audible? Any problems there?
No,
Hello?
sir.
Yeah.
Clear, sir. It's clear, sir.
Yeah. Okay. So let us get into the next of the very important aspect, that Mendel experiments were rediscovered. People are supported. There are other scientists who did the same experiment. They were not able to get the same results what the Mendel or the other contemporary scientists. So that has led to the finally to the understanding more of the mechanism of the transmission of the characters from parents to the offspring, and how genes act together, and that also helped in 1902 the work of the Chutan and Bevere, where they independently demonstrated that the factors of the genes are located on the chromosome, that took the understanding of the mechanism of transmission and the functions of gene further. So that has led to to make a concept of this genetics is the most rigorous and conceptually complete field of biology. So this has made many facets of inheritance. Contemporary scientists working in different modular organisms, they got different kind of results. They interpreted using the Mendel's work as a groundwork. And if the Mendel selected seven characteristic features, where they are not not independently assorted, things would have been different, because all the characters Mendel selected, all of them are present on different chromosome. They could able to segregate as this expectation there. They independently assort them. So the other studies they did, like I will summarize them here, taking very few examples. Interaction of genes. Genes do interact and produce the different kinds of the progeny, and the genes are linked the same chromosome, and even the scientists established sex linkage in the beginning of the 1920. Multiple alleles, pleiotropism, polygenic inheritance, lethal genes, imprinting of genomes. Many new concepts are discovered in different model system that has added, so they as a extension of the Mendelism. So this also the traits controlled by genes, the simple traits, whatever we can make here, dimple, the clipped chin here, the freckle, all the attached earlobe or the separate earlobes, all these are the simple trait, a characteristic feature controlled by genes. this has been, and curly hairs, the straight hair, these are also controlled by the gene or the factor there. So I'll take a couple of examples, only in one or two in each aspects, and try to put forth that how the science has built up. One of the classical example for interaction of genes where two characteristic features have been taken, contrasting features, two genes have been taken. They're supposed to give 9:3:3:1 ratio, but here this interaction, because these two genes were interacted together, they gave 9:3:4 ratio, where both the genes are in a dominant, they give black retrievers. There's the Labrador. I hope people like this example, the phenotype of this, the domesticated dog. There's two dominant genes, they produce a black retriever. One of the gene is dominant, that's a brown retriever. Both the genes are recessive, but however, one of the gene is the dominant. Because of the interaction, they give golden retriever. The whatever the kind of a breed, some of you might have, all that based on this genotypic or the genetic interaction between these two, the color genes. The second example I wanted to quote here with the multiple allelism of the human ABO system. We know that there's Mendel said that every gene has two alleles. Each allele segregated to different gametes, but however, this characteristic ABO system has the four alleles, multiple alleles, but this is the same one characteristic, the blood grouping here. And this also acts as an example for codominant. And we know that the antigens are present on our RBC, the cell, the surface of the cell type. And whatever the antigen is present, that antibody for that agonist will not be produced. Antibody for the other, the protein will produce. In the A group, group A will have the antigen B, antibody B, and the antigen in the red blood cells is here the A. So whereas as we know that group AB group will not produce any antibodies, they will have both the antigen A and B. Similarly, in contrast to that, the O blood group will not produce any antigen, they will have both the antibodies produced. So this is helping to identify the different groups, the clot antigen antibody reactions. I will not go into detail. And now you know that the AB blood group is the universal recipient because it will not produce any kind of the antibody, and whereas the O blood group is the universal donor. Since it's not producing any antigen, it can give into the other blood group. This is the simplest way. People demonstrated, they provided. No, little more the refinement, other blood grouping, other system also taken into consideration. Rh positive, Rh negative, Mn blood grouping, other factors also, but this is the very basic one. So another interesting feature regarding the ABO system, most of you are aware of this. This is the family tree of a ABO blood grouping system. The father is A blood group, mother is AB blood group. However, they gave birth to a O blood group. So then paternity question arises there. So this was proved later by the scientist with the help of the experimental evidences that A antigen and B antigens are being produced with the help of precursor of H antigen. If the H antigen is there, this is the one responsible to make the A antigen and B antigen to move to the surface of the blood cells there. If the H antigen is not present, is in a recessive condition, then the antigen A or B are not move to the surface of the blood cell, that in a default that will become the O blood group. So the precursor for the A, B antigens is very essential. It has to be in homozygous dominant or heterozygous dominant conditions. Then only A and B blood grouping can be seen. if this H antigen is in a homozygous recessive condition, then even though this antigen, the genes or the antigen protein genes are there, they will not be there. So this is called as Bombay phenotype, even though because of the H antigen is the recessive, they are not able to produce that. So this has resolved many of the other paternity tests. Is a very interesting one. One should go to the detailed aspects about this to understand how the whole cascades of the pathways involved to make the antigen productions and attaching to the cell surface. Another example is a sickle cell anemia. It's a classical example. A single nucleotide has changed in the beta globulin gene at the 6th position. A change into T, becoming the glutamic acid to valine. This is well known. And the oval shape of the blood cell become the sickle cell here, which has the defect of carrying the oxygen to the whole body part, the whole body. But this defect in the carrying the oxygen has a many effect on many tissues, many regions like anemia, heart failure, pain, fever, brain damage. All these are because the oxygen is needed in individual tissue. This is not effective. That's why a single gene defect make the multiple effect. That process is called the pleiotropism. So we do have extra finger that is called generally we have 5 the fingers in the hand and the toe and then the leg, and this is all the 6 fingers are normal that develop, that indicating that penetrance and expressivity of all the 6 fingers is 100% here, whereas another here the penetrance is there, the 6th finger, but the expressivity of this is less here, not completely as the earlier one. This is another phenomena. Many of the characteristic features are like this. The human genetic diseases, complete diseases are in this fashion. Penetrance expressivity will not be 100%. If it is a single gene, that will have the 100%, but it's not. Many other genes factors also responsible for this. So our height, weight, obesity, and many hypertension, or many factors are many. This kind of traits are controlled by many genes here. So they are not the abnormal here. They are because of the genomic variation they have. The respective gene, they have different size. Both are normal here. This is called a polygenic trait. So led into the understanding of what is this the factor, the gene responsible. Many scientists have worked with biochemical aspects, molecular aspect. I'm not going to detail account about that. So they identified the characteristic feature of the gene of the prokaryotic genes, and as a unit of function, mutation, and recombinations. And the eukaryotic prokaryotic gene has a simple coding sequences, stop codon, and the start codon, and also regulatory element, and yes, which can give one transcript that can make a polypeptide chain. This is very simple. A eukaryotic gene is not as simple as it is. Eukaryotic genes is a mosaic having exons and introns. Introns are the non-coding sequences within the gene, within the gene, and also this has a regulation of transcription and regulation translation process. And after the RNA is formed, RNA has to be processed and matured to adding the cap at the 5' and the poly A tail at the 3' end to make the stable of this RNA staying in the cytosol for a long time until it gets its activation to become translated to become functional. This is again, I made it in a simple way. It is not so. There's many other factors, many the challenges, complexities also seen in eukaryotic gene. Fairly now we know very well. So another aspect of the eukaryotic gene, even though the gene produce the same protein like mammalian tropomyosin, this protein is differently processed by exon shuffling in different tissues like striated muscle, all these exons will be there. Whereas you look at the brain, exons are the length of the protein, the mRNA will be small here. So different tissue, different kind of isoforms have been formed. They work in that manner. This is a very excellent. So the split gene, the alternative splicing is also possible. Many new concepts have been helpful to understand the mechanism and the gene expression in eukaryotic system. So many functional aspects, functional properties of the nucleic acid, the RNA and DNA has been known: replication, transcription, translation, recombination, mutation, repair, regulations, and also we know that now how the lateral gene transfer from mitochondria to the nucleus, and from nucleus to the chloroplast, chloroplast to the nucleus. This lateral gene transfer is possible. We know several examples. I'm not going to detail about this. This is again, I consider is an extension of Mendelian factor, understanding the gene and the inheritance and their behavior in given set of the organisms. As we know, now we have different types of gene depending on the functionality and also structural organization, like jumping genes, the Barbara McClintock. We have to remember that the genes are they cut and paste into the different region, or copy and paste to the different regions. They bring about the damaging situation of the gene inactivations. Split gene, already said. Edited gene, gene after RNA is formed, then somehow nucleotides added there. Overlapping genes, same genes can be coded for two, same sequences can be coded for two genes. The nested genes, the gene within the gene, and repeated genes. rRNA genes are repeated thousands copies in the oocytes. And process gene to mRNA is transcribed. This mRNA put back to the again to the genome. They are called the process gene, which will have the poly-A tail. And non-coding genes are now making a very big news, that microRNA and non-coding RNA for they are very essential for the normal development regulations of any of the system now, cellular system now. This has led to the understanding of the advent of the technology. Molecular, biochemical, or genetical views have been put forth with contemporary scientists' work, and now it's possible to isolate a gene, insert the gene to another individual, manipulate the existing gene, artificially synthesize a gene. We can make a transgenic animal with the new gene, transgenic plants in the new genes, and also genetically modified foods can be created with the advent of technology. The Mendelian factor, the gene, is gone to that extent. This will have more effect on the society and the humanity. So I'll take another aspects where how well we directly involve our health and the welfare, human genetics and diseases. So human genetic diseases have been classified into 4 types: Mendelian disorders, chromosomal disorder, multifactorial disorders are non-traditional inherited disorders. These genetic diseases are there in humanity about 6%. Chromosomal and Mendelian disorders are very rare, whereas multifactorial disorders are 50% of the 6% is the multifactorial disorders. Even mitochondrial or the non-traditional inheritance of the mitochondria, they are also rare. So as you know, the inheritance autosomal dominant traits, at least one copy of the dominant gene is needed to express that. These are the example: polycystic kidney disease or familial hypercholesterolemia are the example for dominant inheritance of this. And the father has this one copy of that, and one son and the daughter has that because they are in autosomal condition. Very easily explain that. Autosomal recessive disorders, 2 copies needed to be expressed. This we already know that. The example what we know: sickle cell anemia, alkaptonuria, cystic fibrosis, whereas the mother having 2 X chromosomes, this is the autosomal one, and they have to be there in 2 copies to express this one. Another classical example of X-linked inheritance, which has been resolved in the genes are present on X chromosome in 1950s, 1915 and 20 by Morgan, working in the fly as a white fly of the Drosophila, that has extrapolated to the human disease hemophilia and colorblindness. Hemophilia is if you look at the pedigree of the royal family of Queen Victoria, we could able to see that the hemophilia gene A and B are present on the X chromosome, and the female will have two X chromosome. It's very rarely seen in the female, but the male have only one X chromosome. If the mother has the defective hemophilia gene and then contributed to the son, all the son will have the hemophilic one. So this is a very classical example. It has been proved in 1920 itself. Then the Mendelian principles, Mendelian the inheritance was applied directly to the human. People have more enthusiastically started looking into the human characters and the trait in 1920 itself. One of the simple and classical, very profound effect onto the society is that color blindness. Again, this gene is present on X chromosome, and now they were not able to distinguish the red light and the green light in the traffic signals. Sometimes for them, all of them look like the gray here, and they will end up with the accidents there. So this X-linked recessive defect can be corrected now with the corrective glasses. 9% of the society, the humanity has this X-linked inheritance. So I advise or request that people have this kind of the situation. Out of 100, 9 members have this kind of problem around us. So if you have such kind of problem, you can go to the right kind of doctors, you can get a corrective glasses, so that way you can save your life. Human chromosome number was known only in 1956 by Thibault and Levin, of 25 years of the thinking that the human has a 48 chromosome like a chimpanzee. It has been disproved, and they proved with the elegant techniques. And these are the X and Y chromosome. The human has, the male has the Y chromosome, one X chromosome, female has 2 X chromosomes. These are the electron microscopy images. And now there are techniques available to identify any chromosomal abnormalities, aberrations within the chromosome, using different kinds of the binding technique and the fluorescence technique called spectral karyotyping. Is in one chromosome there are 3 or 4 chromosomes segments have been involved. Very elegantly we can explain. This helps to diagnose the cancer patient, or diagnose what is the problem with that and how the situation is there. So molecular cytogenetics is in 1970s taken the shape, and now many diagnoses they use this. As you know, chromosomal the mutations, the chromosomal aberrations, deletions, a part of the chromosome is deleted and that is missing, then the genes which are present in the deleted region will be missing, and it also going to cause the diseases. At duplication, same region, the region is duplicated. While duplicating, the overexpression of 2 copies of the or more copies of the gene will be there. That also overexpression of the gene is also not good, is going to cause the disease. A segment of the chromosome is inverted. Doing so, it will also lose or add certain sequences that also disturb the coding property of the gene present in the vicinity. That also causes the abnormality. Whereas similarly, the translocations, a part of the segment is translocated to other regions. This is called reciprocal translocation. Before translocation, after translocation, that also causes many of the abortion what we see because of the translocated segment. And during the translocation, some certain segments of the DNA or the genes are deleted there. So another very interesting phenomenon is the non-distinction. Has studied where the chromosomes are not segregating properly to the gamete cells. So the abnormal segregation of the chromosome, failure chromosome, causes the very well-known the syndrome called Down syndrome, where trisomy 21 is in the 3 copies. And we did one of the interesting work, very simple work for the Down syndrome. Down syndrome, 1 in 600 in the global populations, and the causative agent is advanced age of mother and non-descent during gametogenesis. But if you compare this in our own, one of the student study of the Down syndrome, that we know that 2 children are born in an hour. Before I complete my talk, 2 children in India born for the young mother, not the advanced age mother. That is the interrogation. Then why young mothers are giving birth to the Down syndrome babies? That was a question we asked long back, 20 years ago. And look at the mother age, or simple the mother age of the Down syndrome as a control mother. Then all the situation we could see. We did analyze the grandmother age of the Down syndrome mother. Down syndrome baby's mother's the age, grandmother age of the Down syndrome kid. We analyzed her age was here as about 35, and grandmother age is 35. Then she conceived the female. That female chances of producing more Down syndrome in India in her young age, or in 25 years, is more. So we made this observations, and we published that also. This has been appreciated and more, that because during the 35, when the grandmother was having 35 years, she is conceived this, the hormonal milieu was not proper, that disturbs for the gametogenesis, which is there in the fetus of the in the germ cells of the daughter there. So this is the link we made. It has been accepted very well there. So multifactorial diseases are having 50% genetic factors, 50% environmental factors, whereas some of these genetic diseases having more of environmental factors, the HIV, and some of them, as I already said, that having the more of genetic factors there. Another interesting factors what we observed in our studies, where we have worked on dyslexia, where the reading and writing disability, we studied about 30-40 families of individuals in the family, and the genetic aspects of the dyslexia we studied. One of the pedigree is this. I hope you can see this pedigree here. Grandfather, after reviewing that, also dyslexic. He was working in a factory. His son was dyslexic. He produced 2 twins. Both are the dyslexic, and these are the ones where we started with this. We started treating this, counseling these 2 kids by one of my collaborator, and then we built up the whole tree. Then easily, this very simply say that there is something like Y chromosome is responsible for this dyslexia feature. We worked very hard. We screened 1.8 million markers for the copy number variation and SNP markers. Finally, we found this in these samples, that one of the gene called PCD11H, that is called protokeratine gene, present on X chromosome and the Y chromosome. Protokeratine gene is the responsible about cell-cell communication. It's a defective. How it defective? These are the red mark, whatever I say, that the gene is duplicated in these cases. Copy number variation, we see this individual, and this has been seen in the X chromosome. So later, when you digged out the information, that we found that there is a region called regions of the XTR, X chromosome transpose regions on the X chromosome. This has transposed to Y chromosome. This is 5 to 6 million years ago, and because the transfer, the copy of the gene is also present in the Y chromosome and the X chromosome, and we did see the recombination between these 2 caused the diseases. I'm making the story short. This is the paper we made, we published, and this again well received. So they generally known that the X and Y chromosome do not undergo recombinations in human. Only terminal region of the X and Y chromosome undergo recombination. They are called pseudo-autosomal region, PAR1 and PAR2. we identify originally, we discovered originally another region between the X and Y chromosome. This is the region we call as part 3. This has been published there. And when doing so, a gene which is present on the Y chromosome, it has gone to the X chromosome and the mother has contributed. And this doing so, that has a copy number varies, and this gene called protochoroidal gene is defective, ended with the dyslexia phenotype. So there are two phenomena: disease gene identification as well as the recombination between the X and Y chromosome we could able to make. This is one of our original contribution and which has been appreciated very well in the scientific world. So my message for the youngsters here is that every observation is unique and important for a new discovery. Any observation, any result you get, you have to record it. If not, that time later we can also resolve it with the right kind of people, interact with the right kind of people. So genetic diseases diagnosis are now very well established, like pre-implantation diagnosis, prenatal diagnosis, newborn screening, and carrier detections are possible. This is help for the couple or the society to go for whether to have an abnormal baby or we can abort it also. This is possible. And control and prevention of genetic diseases is possible. More education is needed, and thank for the organizer now inviting to me to tell that here awareness of genetics is very much needed for the society, for all the people here, the patient, family, and community we need it. And genetic screening, the methods available now, we should know. And also we have management, the groups, the clubs are available for the genetically abnormal kids. This is the way we can control, I can prevent. Even though I have seen the Down syndrome, the child was 25 years old. She is the teacher, dance teacher, whereas making the livelihood with the teaching there. And genetic counseling is very, very important for the family and the individual. Wherever they have that, they will have that. Genetic counseling will help us to estimate the risk and the decision making. And this is the now going on. However, in Indian, the medical school, the counseling is very less compared to the Western medical school. And this is needed now. People are working on it. So the other aspects I will go to take 10 minutes also to tell the newer aspect. What from last 40 years, what we achieved is more than 100 years. What we supposed we would have achieved there. A genome is nothing but the total DNA of the cell, and the genome project has started in 1990 with having a respective specific objectives, and the working draft of the genome sequence, genome project sequence, is published in 2000. This is not 2003. It should be 2001. I think I made a mistake here. February 2001. This has been, but complete sequence of the human genome project was completed on April 14, 2003, and the 13 years they're supposed to do 15 years they completed the 13 years with the public and private funded one. And I hope you know the information. This is very well known. The human genome has 1.5% of the coding sequences, 26% of the introns, and the repeated DNA, remaining of the variety of different kinds of variety of repeated DNA sequences are present. Coding sequence is only 1.5% here. So the haploid cell of the human genome has 3.2 billion base pairs, and we have around 25,000 genes, protein coding genes, and we are identical each other 99.6%, but we are different from 0.4%, that is 4 million base pairs. That is the one making us the uniqueness individually. That is the variation we have about 4 million base pairs between the individual. And the information generated in the genomics is affecting the society by this utility. Genome sequence data now is 1 million copies are available, more than 1 million individuals available. There are very set of data productions are available. Different the companies are sequencing them. data annotations are possible, and interpretation is very, very few people. Some of our very active students, our MSc students and PhD students have been recruited in this kind of company. They are hot cake because the interpreter of the right kind of genetic event or genomic event is essential there. After the genome project, many many varieties of follow-up efforts have been launched. The International HapMap project, 1000 Genome project, ENCODE, synthetic genome, personalized genomics, 1 lakh genomes projects from UK or Asian specific, and genome editing as was possible. I consider this is again from the offshoot of the evolution of the Mendelism in these cases. So now, we are 2020, after 2020, we are heading towards the genomic medicine, gene targeted medicine. We can identify, because almost we are reaching the saturation of genome understanding, genomic variations, understand genomic variation is possible now, and already many companies came forward to identify, diagnose properly, and give the individual-specific therapy. That is called as personalized genomic medicine. Personalized genomic medicine is nothing but the use of information data from the patient's genotype and phenotype to make a strategy for stratify the disease, select the medication, provide a therapy, initiate a preventive measure suited to that patients. This is the personalized genomic medicine. Observation, make variety of tests, take action, and expect the predictable response. This is the breaking the line of the cycle of trial and error medicine, what we are still with this kind of trial and error medicine. So personalized genomic medicine will come very soon. It's already there in the humanity now. In another 5 years, it will be there in our doorsteps there. So diagnosis of genetic disease is very, very essential. Diagnose them, classify them, and that classified group where they are the sensitive for that, you can treat them effectively with the available drug and the new drug, specific drug targeting to that gene. Some of the scientific breakthroughs we know, a sports gene has been identified, ACTN3 genes protein. That protein is needed for fast muscle fiber and which help for the forceful muscle contraction, rapid and forceful muscle contraction, and sprinting and weightlifting. If this is not there, they cannot be one. So now this kind of genes have been are being identified for the sports person, and train them properly. That way they can win. I hope many of you know the Angelina effect. She has the breast cancer and ovarian cancer. The gene called BRCA gene, her mother and aunt has died because of the gene, and she is under the treatment of this, and she is the model for the personalized genomic medicine. Now, genome editing is possible now. One of the Chinese scientists did the genome editing for the CD4 gene, but unfortunately, without the permission, without the ethics, he did it. And though the experiment was successful, he is not been allowed to continue. But now, Russian scientists are eager to manipulate the deafness gene in the early embryo stages where the deafness can be corrected. It is possible that defective gene can be corrected with the concept of genome editing. So to conclude my talk, I hope the 40 minutes of talk will convince you to that genetics is the science of all, where genetics, where the physics, the science, physics faculty can be linked by the radiation genetics, and chemistry can be linked with the biochemical genetics, the biochemistry and biochemistry, and the physiology, and the embryology, where the development biology, where the developmental genes, quantitative genetics, the plant breeding, and mathematics, where without the mathematics, we cannot understand the genetics principles now. Hardy-Weinberg principle, many of the probability we are not able to calculate, and many of the behavioral the problems can be, animal behavioral problems can be understood by the genetics, and the evolution of an organism can be understood by the genetics. Genetics is the center stage where all other science has to utilize, has to apply mind for the betterment of the humanity. So social implications, the genetic profiling and hybrid crops and livestock have societal impact as man's own eugenics. That means he creates his own livestock. Now the crossbreeding of HF and many of the exotic breeds are giving more milks for us. The exotic breed, that is the essence of our artificial breeding and the selecting the gene for the betterment there. Social science genomics is needed because many of to deal with the misunderstanding and misinterpretation of the genetic results cannot be continued. The social science also be involved for the society and the different kind of group involved to understand the genetics and the genomics here. Genomic revolution creates new jobs and improve the economy. Of the Human Genome Project has spent roughly about $4 billion, American scientists, but they got the 5 times more than the economy, the earning for the country because of the instruments, all kind of involvement. And genomic revolution, they also bring about the new jobs and improve the economy of the country. Comparative genomics helpful us to understand the different types of anthropology, cultural anthropology, physical anthropology, and extinct organism that would tell us the history of organism. Many new microbes found in the gut for the human behavior are now. Gut microbiome is the hottest topic now. We also worked on the autism gut microbiome. And that's also very well received now. And people are looking at whatever the behavior we have, it depends on our gut microbiome there. So the genetics help us to plan to create gene edited babies for the deafness. I already told you for the Russia, they're planning to get the ethical clear and so on. The first human paleogenome, using the single hair, they identified the blood type A+, brown eye color, adaptation to the cold climate. This is based on the single nucleotide polymerase, that is genomic variation, whatever they are seen in that is related to this. Now the days are not far away to relate the genotype phenotype relationships. Population genomics for wildlife conservation and management is essential. The analysis of genomic variants of COVID-19, I hope you know, thousands of millions copies of viruses have been, the COVID-19 are sequenced. That has helped our scientists to create vaccines, and India become the world leader. They're creating vaccine in short duration. This is the direction where the understanding about its inheritance, the genomic variation that has helped us. I don't think no one can question of this, the Indian science progress, or the Indian company where the contemporary scientists have contributed for that. Several lessons learned from the genetics, like productive collaborations, basic curiosity-driven research, interdisciplinary approaches are possible by knowing the genetics with the humanities and the engineers, doctors, and the basic scientists together. We can ask many social questions. We can resolve the problems there. So this month in the July, the Nature Review Genetics upheld the same view that, so I quote from the Nature Review Genetics. Genetics have a particular responsibility to engage with the field and public to build genetic literacy, addressing the misconceptions about race, ethnicity, and ancestry, to ensure the responsible use of genetics and genomics information now and in the future. To conclude, the invisible gene was identified, dissected, manipulated, synthesized, and edited. It is no longer a dream. Mendel's principal breeding experiments became an enormously intricate tree of knowledge of genetics. Genetics occupies the center stage in biology and science and meeting the ground for all researchers. Genetics provide the unimaginable wealth for the human welfare and the biosphere. Let us uphold the fire of genetics forever and ever. The take-home message for the learners: genetic diseases are preventable, treatable, and manageable. Genetics should be taught, practiced, and propagated as the foundation of all life sciences. Genomics should be treated as one of the specialized fields like radiology, clinical biochemistry, or clinical chemistry or pathology. Thank you for your patience hearing. So if you have any kind of questions and suggestion and comments, welcome. Yeah.
Thank you, sir. Thank you very much, sir. ಆಡಿಯನ್ಸ್ ಕ್ವಶನ್ಸ್ ಗಳನ್ನ ಕೇಳಬಹುದು. ನಿಮ್ಮ ಮೈಕ್ ನ ಅನ್ಮ್ಯೂಟ್ ಮಾಡ್ಕೊಂಡು ಕ್ವಶನ್ಸ್ ಕೇಳಿ ಒಬ್ಬೊಬ್ರೆ.
Hello, sir. Good evening.
Yeah. Yeah, tell me. Yeah, please.
I have a doubt regarding one genotype of blood hair and hair.
Yeah.
Hello.
Hello. Yes.
Can you hear me?
Sir,
Yeah, I can hear. Yeah.
If the mother is having a blood group A,
Yeah.
and the father blood group AB,
Okay.
how is baby will get this one? How? Because I am a teacher. While teaching, I got this doubt.
How the O group they will get it?
Yeah.
I told you know that this is the concept of Bombay phenotype. A and B, I think that's the way they get it. If A get married to AB, it depends on the H antigen there, how the H gene is working, is the dominant or recessive there. If the H gene, H protein is needed to make the A antigen or the B antigen to move or the transport to the membrane of the blood cells there. If this is not there, even though antigen is produced, it will not able to see. Our antibody will not identify. Only our antibody identify the antigen which is present on the surface of the blood cell, right? This is the way one can prove that. So I think you can read, you can go to the Bombay phenotype or the O blood group phenotype, variation of the O blood group, you can get that information.
Okay, sir.
The one what I presented here, that's also important. So if you go.
Thank you, sir.
Did it clear your doubt?
Yes, actually, I'm not audible clearly. That's why. The voice is too low.
My voice is or your voice?
Actually, in the cell phone. Can you please give your number? I can call you later.
Yeah, it's
ಸರ್ ನಿಮ್ಮ ಮೈಕ್ ಅಲ್ಲಿ ಆಡಿಯೋದ್ರಲ್ಲಿ ಸ್ಪೀಕರ್ ಆಪ್ಷನ್ ಹಾಕೋಬೇಕು. ಕ್ಲಿಯರ್ ಆಗಿ ಕೇಳಿಸ್ತಾ ಇದೆ. ಮೈಕ್ ಮೇಲೆ ಕ್ಲಿಕ್ ಮಾಡಿ ನೀವು ಸ್ಪೀಕರ್ ಆಪ್ಷನ್ ತಗೊಂಡ್ರೆ ಜೋರ ಕೇಳಿಸುತ್ತೆ.
Yes, sir. It's audible now.
Okay.
It is very clear. Yes, sir. Yes.
Yeah. Okay. Yeah.
Okay. Please give your number. I'll tell you later, please.
98800333687.
1 minute, sir.
988.
Yes, sir.
988-900-33-687.
Sir, 988.
Yes. Tell me.
988-900.
No. 988-00 33687.
Okay, sir. Thank you.
Yeah.
Okay.
Yeah.
Next.
Good evening, sir.
Yeah.
Good evening, sir. Sir, actually, I'm also a teacher, sir, and I have one doubt, sir. I was teaching human genome for the children regarding human genome project. Sir,
ಪ್ರೊಫೆಸರ್ ಇಲ್ಲಿದ್ದಾರೆ.
am I audible?
Please mute others.
Yeah, yeah.
Please mute others.
Sir, am I audible?
Yes, you are audible. Yes, ask.
ವಿಡಿಯೋನ ಇದು ಮಾಡೋದು ಅನ್ಸುತ್ತೆ. ಇಷ್ಟೊತ್ತು ಕಾಣಿಸ್ತಾನೆ ಇದೆ.
Sir, can
ಇಲ್ಲ ಏನಾಯ್ತು ಇಲ್ಲಿದ್ದಾರೆ ನೋಡಿ ಇಲ್ಲಿ.
You
ಇಲ್ಲಿ
can ask the
ಬರುತ್ತೆ.
question. It's audible.
ಅವರು ಆಡಿಯೋ ಜಾಸ್ತಿ ಮಾಡಿದ್ರು, ವಿಡಿಯೋ ಇದಾಯ್ತು.
Okay.
Participants request question ಕೇಳೋರು ಮಾತ್ರ ಅನ್ಮ್ಯೂಟ್ ಮಾಡ್ಕೊಳ್ಳಿ, ಮಿಕ್ಕಿದವ್ರು ದಯವಿಟ್ಟು ಮ್ಯೂಟ್ ಮಾಡ್ಕೊಳ್ಳಿ ಪ್ಲೀಸ್.
Sir, Aruna here, sir. Good evening, sir.
Yes.
Yeah, sir, I am teaching human genome project to the children, sir.
Yes.
Regarding that, I was going through the concept. I have seen that there are some places where like centromeres and telomeres and some of the chromatin area where DNA sequencing is not possible. Can you tell me other sequences where it is there, sir, other than these three places where they were not able to study human genome sequencing?
Madam, this is possible now. Even last year or this year only, completely all the 99.99% of the human genome sequence is possible. Even in the repeated DNA in the centromere telomere regions is unraveled now. The latest the informations that they say, databases, we can see that, and we can get that. It's in the beginning when they started, it was not possible. It was only 70%, then 90%, 95%. Now, because of advent of technology, complete sequence is possible now. It is being done now. Not only the human genome project, they have taken 20 individuals, they mix the whole blood piece by piece, the genome sequences. Now, if you give your sample in Bangalore company, within 2 weeks, you can get a complete your own genome sequence, just if you pay around 1 lakh rupees also. It's
Oh,
the
okay, sir.
technology has gone to that extent now. Even if you want to go for exome sequencing, means 25,000 gene, if you want to look, 14,000 to 20,000 rupees, you can get that. The scientists are going to a direction of like a lipid profiling, genome profiling, genome variation profiling also wanted to do. Maybe another 5 or 10 years, it will be less than 10,000 rupees. Because they are using nanotechnology here. The reagent is expensive, but they can use a small quantity, nano quantity to unwrap this. Yes, yes.
So with this, they can study all the genome structure. And one of my students asked me one question that's interesting to know that sequencing is very, very important. What we want to do more about sequence. Suppose if there is a genetical disorder has been there, we are through this lecture I came to know that we can record it and we can do it. Why they need to follow BCR and VCR technique? All these things he was asking me, sir.
Oh, there are n number of techniques are available now. What kind of questions you are asking, for that only we can use the different. Always you need not use the PCR. Okay, now genless sequencing is coming up now. You just isolate your DNA, and there are now nanopore technology for sequencing. That's there. So once you understand once you know the genomes, the sequences, raw data, there are different softwares are there to analyze the genomic variation. There are 4 different types of genomic variation called SNP, single nucleotide polymorphism. Like sickle cell anemia, 1 nucleotide changes, like that. Next one is the microsatellite. Few nucleotides are duplicated or deleted. I would have included that, but the time was less. I not included. Another is the indels, insertion deletion, 100 nucleotide in our genome included, inserted or deleted. The fourth variant is the CNV, copy number variation. The 1 kb, that is 1000 nucleotide to 5 million nucleotide, 5 MB nucleotides are added and deleted in our genome. These are the genomic variation causing the diseases. Other than the chromosomal aberration like deletion, duplication, inversions, and the translocation, what I told you. So one has to analyze where exactly the variation, that the mutation, that the changes are there, which gene is responsible, and what kind of protein the gene is making that. If that protein or enzyme can be supplement, we can supplement and we can rectify it. Such kind of things are coming up now. It's possible now. The genome for example, the Angelina effect I told you, the BRCA gene mutation, BRCA gene, against to that BRCA gene, where the overexpression, a monocular antibody is given to that to suppress that. So suppression is there. The gene is not active in that situation. So there are not many things are there. Unfortunately, our textbooks are revised not frequently. Even revised that also, the latest information not able to added there. That is the one. Maybe another couple of years, everything will rectified there. Lot of
Thank you.
information.
Thank you.
Yeah.
Excuse me, sir.
Yeah.
Thank you, sir.
Yeah.
Sir, Raghavendra here.
Yeah.
Sir, when we look into these aspects, don't you think we are concentrating more on the genomic DNA and we are neglecting the mitochondrial DNA that is connected with so many diseases? Is there any chances that we are having mitochondrial genome sequenced and connected diseases associated with it?
Not really. We are not neglecting even proteomics also. This
Yes,
is the aspect, like when you say genomics, mitochondria also includes, and chloroplast also includes there.
Yes, sir.
Yeah, the nuclear and the extranuclear DNA also includes there. Mitochondria is having only 16 kb and only 32 genes are or 140 genes are there. Yes. Mitochondria and nuclear both are responsible to do that. We are not, the people are ignoring. They even different schools are working on mitochondria. This is the beginning now. Another 50 years, more saturation. Everything will be fished out now with the artificial intelligence, the supercomputers, and the gel-less sequencing, the nanopore technology. Everything will go to be able to understand more about the fundamentals.
Does it mean that the present human genome project also contains the mitochondrial genome analysis?
Mitochondrial genome has been, yes, analyzed. 16 kb is analyzed. They have worked out. Yeah. You see, depending on what kind of disease, they will also going for the mitochondrial genome also, the sequencing.
Yes, sir.
Whatever the present the human genome project, the mitochondrial genome is not included there.
Yes, sir.
The data. But now, even while looking into the family or the pedigree, this is the mitochondrial already known or something different. I think I mentioned there this non-traditional inheritance.
Yes, sir.
A disease is that is not following the Mendelian principle, then we have to rule out, look into the mitochondria genes or the genome or the features of the mitochondrial disease causing features. Yes.
Sir, I have another question, common question, sir.
Yeah.
So if we are interfering too much with the genome editing and other thing, are we not playing with the natural course of evolution that is actually an organism as gifted?
Yeah, certainly. That's why we do have legislations. If the police people are not there, we would have, like we do have our own legislation. Here also, ethical legislations are there. That's why I hope you know that Chinese scientists, the mother was the AIDS patient, and CD4 gene, they rectified, the kids will become negative. Success story. Because he has not taken the ethical consideration. Next day after announcing that in Hong Kong, he has been arrested.
Yes, sir.
Right? Yes. No, see now, uh, one step you can go ahead that you are changing environment, all those things. But now you have money, you know the technology, you have medicine. Who can stop you to go for that kind of technology to rectify our own problems there? That is the questions. In such situations, so genetic defect is known, the mechanism is known, drug is available. Only thing that you have to analyze and you take that. So why you have to stop there? Who are we to stop their right?
Yes, sir.
Not manipulating the whole genome. So even earlier we are talking about cloning, human cloning 10 years ago or 20, 15, 20 years ago. It's when they born, when they become, after 20 years, the environment will be different.
Yes,
Look
sir.
at your environment in the Gangotri, when you were student in Gangotri, when I was student in Gangotri, there were hardly
can't
very
tell it.
few cars. Only two
Yes.
or three cars were there. All faculty were coming by bicycle or by bus or walking.
Yes, sir.
And look at our technology, mobile now, right? So as the things goes, the technology changes, but everyone is upholding that we should not disturb the mother nature, a genome nature. All this understanding for the human betterment welfare. In a sense that if any defective is there, I don't think any baby, any parents are pray for the God or the Almighty that they want a bad gene or a defective gene, right?
Defective. Yes, sir.
Yes. If technology is there to, that is the question they asked when they started a human genome project. 1985, they conceived the idea. Many people opposed that also. Then they said that who are we to stop that? 6 members out of 100 are having defective. Don't you think this is our moral responsibility to make them live equally like others?
Yes, sir.
It's a global notion I'm talking. That is the questions upheld, and that's why the progress in the genomics, proteomics, metabolomics, many other concepts are there. They started with genomics. This is the base. They are interrelated. I'm not telling that this is great or that is great. Yes, understanding of the mechanism is important.
So if we can use it consciously, I think that is a
Yes, we
better
have
thing.
to use it without legislation, without the ethical clearance. If you look at our medical college, wherever we have the ethical committee meeting, every time we will uphold the patients and their family. In nowhere they can create a problem unless they written consent explaining them. They cannot take the sample. Even they want to try a new drug on them also. All these ethical issues will be considered. We're not allowed in our legislation, in our humanity, to kill anybody, right?
Thank you very much, sir. Thank you.
Similarly, we cannot handle the as we wish there. The animal ethics.
Good evening, sir.
All are very, very important now.
Good evening, sir.
Good evening.
Sir, please throw some light on oncogenes, sir.
Yes.
Oncogene. Cancer
You asked the
causing
question
genes.
because I had a limited time.
Yeah.
Oncogenes are the one causing the cancer there. Earlier to that, they are proto-oncogenes, they are the causing. There are genes identified, for example, BRCA1 and BRCA2. They are causing, there are many other genes for breast cancer. So, 2012, UK Prime Minister ordered the whole, the country to go for 1 lakh genome sequencing of the cancer patients who are national health policy patients. 2012. They completed the 1 lakh sequence of 2018, and there are 10 major cancer gene cause the genes that they identified. They are under treatment. UK is going to be a world leader to overcome or to treat the cancer patients. Okay, so they identify the oncogenes, proto-oncogene, whole genome, 25,000 genes. They looked into that. Intricacy of that also they worked out. It's now they are under treatment. From last 4 years, They have started the treatment. 18 centers they established that you go to, if you are interested, the England Genomics. You give the title for the Google search.
Okay, sir.
You can get all the information about how the genome centers in the UK are helping the patients, the national service, the patients there. Okay, so this is, cancer is not something unknown now. Cancer is now only thing that we have to identify in early stages. The respective drugs are available. Many of the many of the breast cancer, the patients have been cured because generally we say the cancer is many genes are involved. Yes, in that particular family, maybe one gene or two genes are involved. We can target to that gene for the treatment. Simply the monoclonal antibody. Similarly, like antigen antibody of blood typing, identifying the blood grouping, the expression of one gene is producing, one gene protein is producing, against to that antibody rise, inject the antibody for 1 week, weekly once or monthly once, depending on the dose, and that the overproduction of this antigen or the protein will be suppressed. Simple treatment. Many of such kind of diseases, hypercholesteremia, the people have studied for giving the statin to them, and 98% are successful. The cholesterol accumulations in the arteries have been reduced in that category of the patient. They did 15 years of studies in UK only. So that's why if you go to go to the heart doctors, they say, oh, cholesterol is more accumulated. They give the heparin or the statin. The thinning of the cholesterol is already the medicine we are taking. It's there in our market, right? So cancer is the first disease where the targeting with the genome sequencing.
Thank you so much, sir.
Yeah.
I am Purandar. Thank you, sir.
Purandar, okay, yeah.
Yes,
Okay.
yes. Thank you so much.
Okay, thank you very much, sir. It's really a wonderful session. Thank you very much on behalf of Mysore Science Foundation.
Okay, thank you for giving this opportunity. It's a good forum. I wanted to be with the forum regularly because I'm also now in Mysore.
Sure,
and,
sir, sure.
This is a good activity. I appreciate. Let's contribute something for the society.
More than 70 participants are participated, sir.
Yeah.
Really, it is a wonderful session and good interaction, sir.
Yeah. Thank you, Santosh.
Thank you, sir. Thank you, sir. Raghavendra, sir, thank you very much, sir.
Thank you, Raghavendra, for inviting me, making me to associate with this. Okay, so we can log out now?
Yes, sir. Yes, sir. Thank you.
Okay, thank you.
Participants, please fill the feedback form, which is available in the chat box to get the e-certificate. Don't forget to fill the feedback form. Feedback form is available in chat box.